So welcome to this session. I'm Minqi Huang from Taipei, Taiwan. I'm currently the treasurer of the CINP. So on behalf of Central Office of CINP, I welcome all of you to go to this lecture. I'm greatly honored to chair this plenary talk, which is going to be delivered by Dr. George Koob. He asked me to make it short, so I'm going to be as brief as possible. Dr. George Koob is the director of National Institute of Alcohol Abuse and Alcoholism, which I've been training there for as a research fellow back then in 2010 to 2011. He is also the senior investigator of the Intraneural Research Program at NIAAA, where he directs the Neurobiological Addiction Laboratory. He's absolutely, no doubt, the iconic figure in drug abuse, drug addiction, alcohol use disorder, and stress. He contributed not only the understanding of acute reinforcing effects of alcohol and drug use, but also he built up the knowledge about the neuroadaptations related to the stress and reward pathways, which is associated with to the transition to dependence. He has published more than 800 peer-reviewed papers before. You know, this is a very astonishing number, which I might need 10 lives to achieve this accomplishment. This is amazing. And he also has authored many books, one of which might be very famous in many countries and might be designated as the textbook of addiction, called Neurobiological of Addiction. I went to the presidential dinner last night, and I realized CINP might be the lighthouse for many researchers. So we are so fortunate that we can get here to enjoy the light from the iconic figure, our speaker today, Professor George Koob. So ladies and gentlemen, please join me to welcome Professor George Koob.
Well, thank you very much. That was really lovely. It's a real pleasure to be here with all of you at CINP. Today, my talk is entitled Alcohol Use Disorder, The Gain in the Brain is in the Emotional Pain and Implications for Treatment. I'm going to start by giving some sobering facts from the United States, pun intended. So we have about 30 million people in the United States with an alcohol use disorder, and we lose about 180,000 people a year to alcohol from deaths that are associated with alcohol ingestion. And half of liver disease deaths in the United States now are caused by alcohol. And I put the population in the United States on there so you can make a comparison to your own country as to the status of the problem, but about 11% of our population meet the criteria for an alcohol use disorder. My favorite quote comes from John Kelly and Sarah Wachman at the Recovery Institute at Harvard. Most middle and high-income countries globally have become largely inured to the endemic premature mortalities related to more commonly used substances such as alcohol and tobacco. While these account for a much larger number of deaths and economic and social harms than opioids each year, the devastation wreaked by these substances, their casualties, and the associated blood and tears are all relatively willingly absorbed into the social fabric. So I don't deny that we have an opioid overdose crisis in the United States. For those of you who don't have an opioid overdose crisis, you should praise somebody. But the fact of the matter is we have an endemic problem with alcohol, and most countries in the world, according to the World Health Organization, also have an endemic problem with alcohol. So what am I going to tell you today? Well, I'm going to tell you three things. Individuals with alcohol use disorder are miserable. Individuals with alcohol use disorder suffer a hypersensitive negative emotional state during acute and protracted withdrawal, a state that's called hypercafia. Neurobiological substrates mediating negative reinforcement in addiction provide insights into additional sources of motivation for compulsive drug seeking. The brain circuits that drive such a negative emotional state, hypercafia, in addiction are key to understanding the role of allostatic loads that contribute to the etiology and perpetuation of alcohol use disorder. Now, I'm going to explain all these words that I've just thrown at you, so stay tuned. But I want to also point out that I could give this exact, in fact, I will, 2 days from now, give this exact same lecture with opioids. So what I'm trying to get across today is there's a universal component to this framework that I think some of you may find useful. So what I'm going to talk about is alcohol use disorder or overview of origins and validation of the three-stage, three-domain framework, the definitions and heuristic conceptual framework for understanding the negative emotional side of alcohol use disorder, opponent process, hypercafia, hyperalgesia, and negative reinforcement. I'll talk about alcohol use disorder and hypercafia and its neurobiological substrates, the convergence of pain and negative emotion neural circuits, and the implications of that, protracted withdrawal, or what I coined recently, protracted hypercafia, implications for relapse, treatment, and recovery. And I'm going to end with, assuming I can get through all this, I'm going to end with social determinants of health, deaths of despair, COVID-19 pandemic, driving alcohol use disorder via negative reinforcement, and the implications for treatment. So let's get started. So how do I define addiction? Well, I define addiction like the rest of you. Chronically relapsing disorder characterized by a compulsion to seek and take drug, loss of control and limiting intake. But there's a Koob add-on, which is the emergence of a negative emotional state, dysphoria, anxiety, irritability, which we define as hypercafia, when access to the drug is prevented. So what is the three-stage cycle? Well, it's actually the three stages that are involve three domains of dysfunction and three neurocircuits in the brain. So the binge intoxication stage engages incentive salience and pathological habits and is mediated by largely circuits that involve the basal ganglia, which are in blue on this diagram. The withdrawal negative affect stage engages reward deficits and increases in stress and involves neurocircuitry that's in red here, which is largely the extended amygdala. And the preoccupation anticipation stage, if you prefer craving stage, involves executive function deficits and is mediated by largely cortical projections to these subcortical circuits. So largely frontal cortex, allocortex like hippocampus and insula. Where did it come from? Well, there's lots of things with me. I made it up, all right? I was teaching a course at the University of California, San Diego on impulse control disorders, and I discovered that not only did drug addictions, each drug addiction, follow this same pattern of three stages, but so did non-drug addictions, or what the American Society of Addiction Medicine calls process addictions, like gambling or excessive eating or compulsive-like eating, rather, or compulsive exercise. And so when I put the course together, you can see it was in 1997, and the fourth item down is addiction cycle, and it was this addiction cycle. And then Michel Lemoyle and I published a paper in Science where we superimposed the DSM-4 criterion at that time on these three stages. And the rest is kind of history. Many, many people, many, many laboratories contributing to the overall picture. When I came to NIH in 2014 and began interacting with our staff in the intramural program, because I'm also part of the intramural program, as was explained, I have a laboratory at the National Institute on Drug Abuse, but I direct the National Institute on Alcohol Abuse and Alcoholism. Don't ask me why, all right? NIH, all right? But we all got together with a number of intramural researchers led by Laura Kwakow and David Goldman and Ray Litton from NIAAA, and we asked the question, could we provide a framework using the three-stage cycle that would improve diagnosis, prevention, and treatment? And we called it the Addictions Neuroclinical Assessment. And the goals were to identify how the three domains reflect differences between people diagnosed with alcohol use disorder, which then could be used to guide treatment decisions, and to better understand the differences between individuals with and without alcohol use disorder. So we eventually, there's a protocol underway, but eventually Laura Kwakow went back to 454 patients that passed through NIH's clinical center, and we looked at everything from hedonic scales and different scales for emotional changes to things like delayed discounting. But we also looked at some neuropsychological tests, like the cue reactivity test for incentive salience and facial emotion matching tests for negative emotionality and delayed discounting task for executive function. And what we found was, doing a major factor analysis, was that the three domains of dysfunction that fell out were, in fact, incentive salience, negative emotionality, and executive function. And subsequently, there are 6 papers in the literature from laboratories across the United States doing exactly the same thing with different cohorts of subjects. And it's always the same 3 domains that fall out. So if you accept these 3 domains, then putting it in a perspective of how does addiction transpire, I'm not denying that there is reward, all right? It's very important. That's how you get started. But reward shows tolerance with addiction, and particularly with alcohol use disorder. It even happens within one session of drinking. So think about that first drink versus the 4th drink. And I don't recommend the 4th drink, just for the record. Then you get into, and you move into, as you keep drinking, another source of reinforcement, which is called relief. And then, maybe you do stop. Maybe you've been through a binge and you stop. Or maybe you have an alcohol use disorder, a moderate to severe alcohol use disorder, which would be similar to what we used to call, or what ICD-10 or ICD-11 calls alcohol dependence. Then you may go into a protracted abstinence, and ultimately you relapse. But one of the interesting things about this diagram that was originally put together by Larry Parsons, who was at Scripps Research, passed away prematurely, but he had a loop back to the relief stage. And so any of you that have done animal studies know that once an animal is quote unquote addicted, they return to that addicted-like state very rapidly. It's not like they start over again. And one of the points I meant to point out is that you can enter the addiction cycle at any one of the stages. People think that, well, you have to go through the binge intoxication stage. You don't. The opioid crisis points out very vividly that you can enter through the withdrawal negative affect stage. And what about somebody with attention deficit hyperactivity disorder who's taking drugs to control their impulsivity? They're entering through the preoccupation anticipation stage. So why is this important, these 3 neurofunctional domains as a framework for alcohol use disorder? Well, one, and this is putting on my director's hat, it reduces stigma. Alcohol use disorder is considered a brain disorder that can be treated. It individualizes diagnosis and treatment. We hope it will facilitate treatment based on individual differences in which domain one enters the addiction cycle. We hope it facilitates recovery, provides a framework to help clinicians focus on the different triggers for relapse. We hope it facilitates prevention, provides a framework for identifying individuals with domain-specific vulnerabilities, both genetic and environmental. And just a quick example that I'll elaborate on just a little bit more later, high scores in the negative emotionality domain showed significant associations with more frequent and heavier drinking, and drinking to regulate negative affect. And that was in a fairly recent paper. So what about all these words that I've been throwing out and the heuristic conceptual framework for understanding hypercafia? Well, this is my favorite painting in the world, practically. It hangs in the Hermitage Museum in Russia, as I understand it. I've seen it twice, once in Paris and once in New York. It's the Absinthe Drinker by Picasso. If you look at all the absinthe paintings of the turn of the century, from the 19th to the 20th century, 18th to the 19th century, rather, in the post-impressionist era, you'll be hard-pressed to find one of them where someone is smiling. And absinthe was a very potent alcohol-containing beverage, included also a stimulant. So I'm going to focus on the withdrawal negative affect stage. And a lot of my thinking came from Richard Solomon, who I met just once, many, many years ago at the University of Pennsylvania. But he argued that and he was using opiates for this paper that was published in 1980. He argued that there was an A process, which is basically getting high, getting intoxicated if you prefer alcohol. But when the high wears off, and it does within one session for alcohol, as I pointed out, that's called within-session tolerance, there's often a rebound, which is called a hangover with alcohol, a rebound in the negative direction, or what I would call a mini withdrawal state. But if you keep drinking, or you keep taking opioids, as he illustrated, you lose the A process, and in fact, you gain the B process, and there were arguments like from Francis Colpert years ago that the B process was actually subtracting out the A process. Now, you don't sit around at night obsessing about what causes a B process, but basically the last 25 years of my life have been dedicated to trying to understand what causes that B process. A very simple way of putting it is hedonically, what goes up must come down. So I got tired of writing and saying hypersensitivity to a negative emotional state, and I made up a word, all right? I called up my brother Steve, and I said I need a Greek word for rejection, dejection, sadness, or negative emotional state, so he gave me katifia. I stuck hyper on it, and it's defined as the increased intensity of negative emotional, motivational symptoms and signs observed during withdrawal from drugs of addiction. It's hypothesized to represent elements such as dysphoria, irritability, alexithymia, or simply symptoms often described as ill at ease, uncomfortable within one's own skin, or simply not hedonically normal, symptoms historically difficult to define. And since many of you are psychiatrists, I'm not talking about major depressive episodes here. I'm talking about an underlying negative emotional state that is less dramatic than a major depressive episode. And it's paralleled by another term that is prominent in both alcohol addiction and opioid addiction, and that's drug-induced hyperalgesia, which is defined as a state of diffuse increased pain sensitivity to a stimulus of stable intensity observed during withdrawal from drugs of addiction. And of course, I was thinking of hyperalgesia when I coined the word hypercafia. It's in Wikipedia, you can find it now. I'm very proud of the fact that it exists as a word. It wasn't an easy sell. When we submitted the paper, I did this with two anesthesiologists, Joe Sherman from Scripps in California, Scripps Hospital, and Howard Goodstein, who at the time was in MD Anderson. The reviewers came back and said, hypercafia doesn't exactly roll off the tongue. Well, most of the time you give in to the reviewers, but I wanted my word in literature. So I sent back the paper and I said, hyperalgesia doesn't roll off the tongue either. So it got published. So it's out there. All this is predicated to another source of reinforcement that I would argue has been largely neglected in the addiction field. And maybe not so much anymore, because I think we're starting to understand, and I'll illustrate later how the COVID-19 pandemic actually brought home this message. But another source of reinforcement in addiction is what we call negative reinforcement. And that's defined as a process by which removal of an aversive stimulus, and I'm going to argue the negative emotional state of drug withdrawal, increases the probability of a response, dependence-induced drug taking. Negative reinforcement is not punishment, Punishment in psychology is the suppression of behavior by presentation of a stimulus. Negative reinforcement is where you increase the probability of a behavior by removal of something aversive. It's critically important when you come down to some of the issues that I'm going to be talking about. So how does this fit in with allostasis, opponent process, hypercafia? How do they all fit in negative reinforcement? Well, I'm going to take you through this and then I'm going to elaborate. But because of an opponent process, a greater quantity and more frequent use of the previously rewarding drug are needed to maintain or approach euthymia, normal mood state. That's called tolerance. It's not a bad word. It's one of the criterion for, in DSM-5 for an alcohol use disorder. When the substance is not used, then negative emotional signs of withdrawal emerge, such as irritability, anxiety, dysphoria, sleep disturbances, and subjective feelings of need. This deficit emotional state, or hypercafia, sensitizes and becomes persistent with repeated use. And we call this now protracted hypercafia. Under this conceptualization, drug intake is compulsively escalated or renewed in relapse via negative reinforcement processes because the drug transiently prevents or relieves this hypercafia. Drug taking for relief ultimately becomes, though, self-defeating because it worsens and perpetuates the vicious opponent process cycle, creating hedonic allostasis. What is allostasis? It's stability with change, where you change your set point. It's a break with homeostasis, but you haven't quite reached pathology yet. And so ultimately, as I say to lay audiences, you end up drinking to fix the problem that drinking caused. So how did you get there? What happened in the brain to create hypercafia? Well, a long time ago, Floyd Bloom and I conceptualized that there were two types of adaptations that could exist in addiction. This is actually a science paper in 1988, believe it or not. And we called these a within and a between system neuroadaptations. A within system neuroadaptation we defined as a process by which the primary cellular response element to the drug would itself adapt to neutralize the drug's effects. But a between system neuroadaptation, and if I may say so, I think we were a little prescient with this, was defined as a process by which a different cellular system and separable molecular apparatus would be triggered by the changes in the primary drug response neurons to neutralize the drug's effect. So you all know, presumably, that there is a within-system neuroadaptation with all major drugs of addiction. It's our beloved dopamine system. So ethanol withdrawal decreases dopamine levels in extracellular fluid of the nucleus accumbens using in vivo microdialysis. This was a beautiful study by Marco Diana, and many others have shown this over the years, and also decreases firing of the VTA dopamine neurons, which is on the right side of this diagram. But in a kind of really clever study done by Bert Weiss at the Scripps Research Institute, he showed that ethanol self-administration, where the rats could self-administer the alcohol themselves orally, they pressed a lever, they drank the alcohol, that reversed withdrawal-induced decreases in dopamine dialysate levels. And you can see that in this slide where during withdrawal, there's a dramatic decrease in dopamine in the extracellular fluid of the nucleus accumbens, but the animals, when they're allowed to self-administer the drug, actually return it back to the normal level, at least temporarily. And of course, many of you know the classic work of Nora Volkoff, and she has shown over and over again that there's a decrease in D2 receptors that's constituently found in individuals with drug addiction. But she's also shown with alcohol and with opioids, or others have shown with opioids, that if you challenge the subjects with methylphenidate, you see no decrease in binding of the D2 receptor, indicating that there's a blunted release of dopamine in the individuals with alcohol use disorder. That's what these data show. So the control subjects show the decrease in binding, which means the release of endogenous dopamine. The alcoholic subjects or alcohol-dependent subjects show no such decrease because they're already, hypothetically, in a deficit state. What about between-system neuroadaptation? Well, there's a whole group of neurotransmitters that we have now identified, most of them localized or projecting to the extended amygdala. This diagram shows the extended amygdala in red on the right-hand side, but it also shows a blow up the extended amygdala with many of these neurotransmitter systems converging on the extended amygdala, which includes the bed nucleus of stria terminalis and a transition zone in the shell of the nucleus accumbens. I'm going to focus on three of these, corticotropin releasing factor, dynorphin, and glucocorticoids and mineral corticoids. And I'm going to show you I'm going to actually show you data, and I'm going to show you some new data from our laboratory on glucocorticoids and mineral corticoids that we find quite intriguing. But there is data for all of these neurotransmitters, and I want to point out, and I'll try and make this point again, there are also buffers to the stress neurotransmitters. So you can see the list here that include vasopressin, hypocretin, substance P, and neuroimmune factors. But their buffers to this include neurotransmitter systems such as neuropeptide Y, nociceptin, endocannabinoids, and oxytocin. One more little vignette. If you step out in front of the traffic here in Tokyo, not looking the right way because they drive on the wrong side of the road. I didn't say that. Maybe they drive on the correct side of the road. But in any event, and then you realize that you've stepped into traffic, you're going to do one of two things. You're going to either run across the road or you're going to freeze. But you're going to notice when you're accomplished saving yourself from getting squished that you suddenly can breathe again, and that your heart rate's up and your blood pressure's up. And if you were thinking about your next experiment, it's now clear exactly what the experimental design is. That's the stress system that I'm talking about. We need it, we use it. But in addiction, it gets overworked and overextended and sensitizes. And that's going to be the theme here. So I was actually involved in the early stages of characterizing the physiological actions of corticotropin releasing factor. I was at the Salk Institute when Wiley Vale and Joachim Spiess and Catherine Riviere and Jean Riviere sequenced CRF. And over the years, there's been a whole series of studies, mostly in rodents, that have shown that CRF seems to play a very important role in mediating hypercafia-like behavior in rodents. And so we know there's an increase in CRF expression in the amygdala during withdrawal from alcohol, and we know that CRF antagonists can block withdrawal-induced responses to alcohol, can reverse dependence-induced drinking, and can reverse stress-induced reinstatement. Some old data from our laboratory, that's one of the more compelling things we ever did. We microinjected a peptide CRF antagonist into the brain bilaterally, into the amygdala, of rodents that were dependent on alcohol in the white bars and not dependent on alcohol, or we called them social drinkers, in the bottom bars. And you can see that the CRF antagonist reversed this compulsive-like alcohol intake. We've gone on to many studies. This was a more recent one by Giordano Di Guglielmo and Olivier George using CRF Cree rats that Robert Messing made. And we finally published this in 2019. But it showed that inactivation of a CRF amygdalofugal pathway reversed dependence-induced drinking in the rat. So in this case, there are FOS-activated neurons in the central nucleus of the amygdala during withdrawal, and they're accounted for, 80% of them have CRF in them. And so what we found is if we used halorhodopsin and a viral vector to convey the halorhodopsin, which would decrease firing of those CRF-specific neurons. So each brain region was targeted using halorhodopsin expressed in CRF terminals or CRF neurons. The only regions that replicated the findings obtained with CFOS-activated neurons and CRF antagonists was this central nucleus of the amygdala to the BNST pathway. So optogenetic inhibition of central nucleus of the amygdala CRF neurons when the laser was on, which is the light shown on to activate and inhibit CRF neuron projections in the bed nucleus, decreased alcohol intake. But it didn't work in the lateral hypothalamus, the substantia nominata, or the parabrachial nucleus. So, you know, it looks like there is a circuit between the central nucleus of the amygdala and the bed nucleus stria terminalis, and it may be actually a reciprocal circuit that involves CRF that is key, at least in rodents, in driving excessive alcohol consumption in the dependent state. But I had a postdoctoral fellow at the time named Leandro Vendroscolo, and Leandro kept saying that we need to look at what modulates the CRF system. And he said, you know, we should look at glucocorticoid antagonists. And I said, Leandro, you know, that's a separate part of the system. That's all peripheral. It doesn't have anything to do with the brain. But Leandro insisted, and then we went into the literature and we discovered that chronic glucocorticoid activation blocks further activation of the HPA axis, the hypothalamic pituitary adrenal axis, by blunting CRF mRNA in the paraventricular nucleus. You all got that information in medical school, probably, those of you who went to medical school or physiology courses. So if you give chronic glucocorticoids to rodents, corticosterone exposure, you blunt the expression of CRF in the paraventricular nucleus. I often say that's why in the United States, if you're writing a very difficult grant, why don't you get Cushing's disease? Well, because you have this automatic mechanism to suppress further release of glucocorticoids. But I also say you still snap at your significant other. You're still irritable and cranky and not a very pleasant person to be around when you're writing a big grant. And that's because the glucocorticoids feed back to the amygdala and they drive CRF expression. And that also occurs for alcohol. So you see a blunted CRF expression with chronic alcohol in the paraventricular nucleus, but you see a sensitization in the central nucleus of the amygdala. And I might add, there's exactly the same results with opioids. So we went on to do a whole series of studies, and most of this is recent work, and I'm going to go through it very quickly. But we found that glucocorticoid antagonists decreased dependence-induced drinking in rats. That's what's shown at the top there. We used the drug mifepristone, which is also a progesterone antagonist. We microinjected it into the central nucleus of the amygdala. We got exactly the same results. And then, because it's a progesterone antagonist, we also used a specific glucocorticoid antagonist given to us by Corcept, and that also produced the same decrease in alcohol drinking. And Barbara Mason, at the time, was looking at mifepristone in a human laboratory study and was able to show that it blocked craving in human subjects, which is shown on the bottom here, but it also had this residual effect in decreasing drinking for 2 weeks after the initial exposure for 2 weeks of mifepristone. So we've gone on to look at this further. And I just wanted to show this slide to emphasize to you that those original studies were all done in male rats, but we've replicated them in female rats, and this is the female data. It's unpublished data from a post-baccalaureate fellow, Valerie Acosta. And during this period, in collaboration with Mehdi Farahkhania and Lorenzo Leggio and many others, we asked the question, what about a mineralocorticoid receptor antagonist? Would that have similar effects on alcohol drinking? And there was a clinical reason for doing this. And you can see here, both in male and female rats, that spironolactone, which is a mineralocorticoid antagonist, it's a medication that's used for everything from high blood pressure to electrolyte imbalances. And in addition, if you skip down to the last bullet here with me, the use of a mineralocorticoid antagonist prescription in humans was associated with a greater reduction in alcohol consumption compared to propensity score matched individuals who did not receive spironolactone. This was what we call in the business now a pharmacoepidemiological study with a large cohort of individuals, and there have been 2 of these studies now, and in both cases, spironolactone taking individuals were drinking less than non-spironolactone taking individuals. So we're quite excited about these results. There are ongoing clinical trials and planned clinical trials in both of these domains of modulating the HPA axis interaction with the central nervous system stress system. And I just want to point out to you, you could say, well, how is it that glucocorticoid receptor and mineralocorticoid receptors could interact? Well, they both have genomic effects that are slow, but they also have transcription and rapid. They're slow via transcription, but they also have non-genomic effects, which are rapid by cell signaling. And there's even a hypothesis, which is on the right side of this slide, that steroid hormone receptors can possibly also interact through the ability of GR and MR to dimerize with one another in the nucleus. So these are sort of, for us, exciting times with a lot of potential for understanding, where in the brain is the fire when it comes to the CRF hypothalamic pituitary adrenal system. And some of you know that CRF antagonists have not had a real big success in psychiatry given to humans. Limited work has been done in the addiction domain. But maybe, as Alan Schatzberg said, we're trying to put the fire out in the wrong places. We should be putting the fire out in both the pituitary and pituitary adrenal system and the central nervous system extended amygdala. Food for thought and translation. I'm not going to spend time on all the data that support what's on this slide, but let me just argue with you that we have exactly the same pattern with kappa receptors and the dynorphin kappa system. It's activated in the nucleus accumbens shell and in the extended amygdala during withdrawal from alcohol and other drugs of addiction. And kappa opioid antagonists are being shown more and more to block hypercafia-like responses. And many people think that that is because it actually not only blocks stress-like responses in the extended amygdala, but may be restoring dopamine function because, in fact, kappa agonists suppress dopamine function, as shown in this elegant diagram from Eric Nestler and Bill Carlazon many years ago. So I'm back to my diagram. I just reiterate the same point I made earlier. A between neurosystem adaptation that mediates hypercafia and negative reinforcement may be activation and sensitization of this brain stress system in the extended amygdala. What about pain? I'm talking about emotional pain, but what about pain in human beings? So we do congressional briefings every once in a while where our staff go down and colleagues go down to the Congress in the United States, and we talk basically to congressional staffers. Sometimes a congressman or a senator shows up. And we did one on women and alcohol, and Martha Woodruff, who passed away subsequently, gave a beautiful talk about alcohol in her personal life. And I asked her for a quote, and she sent me this. "I grew up professionally focused and personally adrift. There was a long history of depression in my family, and my childhood home was dominated by my mother's mental illness that was not recognized, let alone treated. Sometime in my mid-30s, I started using alcohol to provide relief from the pain of childhood damage and depression. Bourbon was the only thing that would make the loop tape of despair in my head pipe down." We know alcohol reduces pain. It takes a lot. It's basically a binge level, 0.08%. This cartoon says, "I don't prescribe opioids anymore, so have your bartender fill this prescription." But this could explain why people in chronic pain often misuse alcohol despite the potential consequences to health. We know that more severe alcohol withdrawal and higher depression scores are associated with greater pain sensitivity. And so the left-hand diagram here shows that subjects undergoing alcohol withdrawal show increased pain sensitivity, but there's decreased mood of patients was also associated with reduced tolerance to pain. So higher depression scores on the Beck Depression Inventory were associated with more sensitivity to pain in a one-to-one kind of relationship here in this diagram. Where would pain overlap with the addiction circuitry? Well, of course, the circuits for pain involve, the classic spinal thalamic pain pathway, but also the parabrachial directly to amygdala pain pathway. And of course, they overlap in my beloved extended amygdala. And just to kind of put a wrap on this relationship, we know that animals will show hyperalgesia. And in using a Paul withdrawal reflex measure, a number of researchers have shown that during alcohol withdrawal, and you can see it here, this was done by Scott Edwards when he was a postdoc with me, there is a hyperalgesic response. We could reverse that partially by administering systemically a very effective CRF antagonist. So you see the crosstalk. And in fact, Victor Nojbauer has done a beautiful seminal series of studies showing that CRF has a critical role in the aversive, affective, and sensory pain behaviors. And you can see here that the nociceptive input reaches the amygdala through the lateral parabrachial area. I showed that in the previous diagram in the brainstem, but it targets PKC delta neurons, somatostatin neurons, and CRF neurons. They're a prominent part of the nociceptive amygdala. And polymodal information reaches the amygdala from sensory cortical and thalamic areas via the basal lateral amygdala, which is diagrammed on the right-hand side of this diagram. So the place and even the molecular apparatus where physical pain and emotional pain interact is very possibly in a very specific part of the medial and lateral amygdala. And I always say, again, to lay audiences, physical pain causes emotional pain, and emotional pain causes physical pain. And you can see that this structure that's heavily involved in addiction from the negative reinforcement perspective is also very possibly involved in how we interpret pain. And in fact, the catastrophizing anxiety, negative urgency, and expectancy model that Jeff Bolsonault and his team have been writing about involves negative affect as a mediator of excessive alcohol use, alcohol misuse, with intervening variables like negative urgency, which is the disposition to act rashly under distress. Remember running across the room that the road that I talked about. And so again, pain can directly cause alcohol self-administration behavior to relieve the physical pain, but negative affect amplifies that, and that's my point. What about protracted withdrawal? Well, this is a diagram I stole from Barbara Mason. She's always argued that the preoccupation anticipation stage involves more than just cues. There's also an underlying negative emotional state that continues into the preoccupation anticipation stage. It's been called protracted withdrawal. I coined the term last year at Volterra as protracted hypercafia. You can use whatever term you want. But the fact is we know that protracted withdrawal or protracted abstinence can be defined as withdrawal symptoms that persist past acute withdrawal phase that follow the binge intoxication stage and continue through the preoccupation anticipation stage. For patients with AUD, protracted withdrawal begins approximately 1 week after resolution of physical withdrawal symptoms. Symptoms include hypohedonia, anxiety, and sleep disturbances, and typically abate within 3 to 6 weeks, but can persist longer in a more subtle form. What we see in rodents is that the animal may appear normal, but if we put them in a stressful environment, then they have an exaggerated response to stress that is uncovered with a history of alcohol dependence. Early work by Alan Marlatt showed that negative affect stress during protracted withdrawal contributes significantly to relapse in alcohol use disorder. The leading precipitant of relapse in a large-scale replication of Marlatt's taxonomy analysis was negative affect. And anhedonia is a key component of protracted abstinence. Marlatt's tables show about 2/3 of people were actually showing relapse to a negative emotional state of one form or another. And actually, if you go back to Mark Schuckett and Sandy Brown's early study in 1988, we're using the Hamilton depression scores. You can see that during acute withdrawal, the subjects never made it to a full-fledged clinical depression where you would prescribe perhaps an antidepressant drug. But even 4 weeks later, even though there's a decline in the Becker depression score, the Hamilton depression score, they're not tiptoeing through the tulips. And I found a remarkably PhD thesis from the University of Adelaide that I can't find any subsequent publication of, which is an identical replication of what Sandy Brown and Mark Schuckert published in 1988, and that was in 1999. And now to put on my NIAAA hat one more time, we've decided we needed an operational definition of recovery to advance treatment on research on treatment. Most people who need treatment receive no treatment of any kind in the United States, and little is known about what sustains longer-term recovery. So we expanded are expanding our focus on long-term recovery. So we define recovery from alcohol use disorder based on qualitative feedback from key recovery stakeholders, researchers, clinicians, and recovery specialists. We view recovery as both a process of behavioral change and an outcome that incorporates time points for two key components: remission from DSM-5, alcohol use disorder, and cessation from heavy drinking. This is a non-abstinent recovery outcome. Some would argue it's a harm reduction-like recovery outcome. The NIAAA definition of recovery also emphasizes the importance of biopsychosocial functioning and quality of life in enhancing recovery outcomes. And just to bring this home, Katie Witkowitz's group has been doing a whole series of studies, and she shows that that greater relief negative emotionality at baseline predicted greater drinking intensity and more frequent heavy drinking during recovery. These are beta scores, which is the degree of change in the outcome dependent variable for every 1 unit change in predictor variable. And you can see the only one that was really significant was the relief negative emotionality. This particular study validated again the 3 domains of the 3-stage addiction cycle. And these domains of the addiction cycle, interestingly enough, were more strongly associated with outcomes than the AUD symptoms themselves. I thought that was striking. And using measures from Project Match and Combine, the results of this study supported the utility of the domains of the 3-stage cycle in predicting AUD treatment outcomes and recovery 1 year post-treatment. At 1 year follow-up, relief negative emotion scores were associated with drinks per day and percent heavy drinking days initially. I'm going to end with a brief discussion of the social determinants of health. Some of you may have seen this diagram that I put together years ago with Michel Lemoine. It comes from Sterling and Ayer's hypothesis about allostasis and stress. You can see here the opponent process, and our argument is the opponent process never really disappears. But what happens is there's a drift below baseline, below a certain normal hedonic set point. And the other argument from this diagram, and allostasis here is considered a manifestation of allostasis is this altered hedonic set point, all right? I'll repeat that. The manifestation of allostasis is an altered hedonic set point, stability with change. But weighing down this drift in set point is genetic, epigenetic factors, childhood trauma, psychiatric comorbidity, that's for you all, and excessive drinking. And you can get there in any one of these different ways. Don't forget, you can get there by drinking a lot. We're interested at NIH in a variety of social determinants of health. In my field, we're particularly interested in its impact on alcohol misuse and alcohol use disorder, and we're going to argue that they could also be weights on this allostatic process, allostasis process. So social environment, discrimination, racism, social isolation, growing up at home with a parent with alcohol use disorder, physical environment, alcohol outlet density, exposure to violence, poor health care services, access to and quality of care, economic instability, lack of job security or stable income, limited education access and lower quality, lack of educational opportunities and support, adverse social determinants of health serve as allostatic loads on the body's stress system, increase vulnerability to mental and physical health conditions, and contribute to health disparities and inequities. Such stressors can drive alcohol misuse to cope, which in turn exacerbates the initial problems, further fueling alcohol misuse. We wrote this paper at the peak of the pandemic, and the pandemic really, as I said earlier, shined a light on these issues about stress and coping and drinking to cope with stress. And so during the pandemic, people were worried about their jobs. People were worried about getting sick, particularly the elderly population. People had more obligations at home because the kids weren't in school, in the United States at least. And furthermore, they were isolated. And isolation has turned out to be a major stressor to human beings, as you might expect. All of this exacerbated this impact on the withdrawal negative affect states that we associate with compulsive drinking or compulsive-like drinking. And, you know, I might point out to you that in the first year of the pandemic, we had a 25% increase in deaths associated with alcohol marked on death certificates in the United States. We had another 10% on top of that the second year of the pandemic, and it hasn't come down yet. So this is what I told you. Alcohol use disorder represents a dysregulation of incentive salience, negative emotion, and executive function domains, corresponding to a dysregulation of neurocircuitry with focal points in the basal ganglia, extended amygdala, and prefrontal cortex, respectively. Individuals with alcohol use disorder are miserable. Individuals with alcohol use disorder suffer a hypersensitive negative emotional state during acute and protracted withdrawal, state called hypercafia. Neurobiological substrates mediating negative reinforcement and addiction provide insights and additional sources of motivation for compulsive drug seeking. And I might also add, they provide insight in how emotions are processed in our brains in the first place. Such within and between system neuroadaptations include loss of dopamine function, activation of the CRF, hypothalamic pituitary adrenal axis, and other brain stress system neurocircuitry. Withdrawal from alcohol causes pain, both physical and emotional pain. Social determinants of health interface with addiction as allostatic loads to the brain negative emotional systems. The brain circuits that drive hypercafia and addiction are key for understanding the role of allostatic loads that contribute to the etiology and perpetuation of alcohol use disorder. So the next few slides are just summary of that. The left-hand side shows the hypohedonia. The right-hand side shows the hypersensitivity and hyperactivity of the stress systems. There are numerous targets now out there in preclinical work and beginning to be in clinical studies and human laboratory studies that have been identified that target these specific elements of what I would call the between-system neuroadaptations in addiction. And I've listed some of them here. You can find them in this review article. I already gave you the 3 take-home messages, so I won't reiterate that. This is my laboratory at the National Institute on Drug Abuse. We have a bunch of wonderful post-baccalaureate fellows. These are individuals who had a university degree and are going on to medical school or graduate school, and I'm proud to tell you that every one of our post-bacs goes on to medical school or graduate school. I had a great staff scientist, but he just got promoted to his own unit, Leandro Vendrascolo, but I have a new staff scientist, Renata Marchette, and who's also excellent, that helps coordinate all the elements of the lab. We are part of what we call the Koob Leggio Vendrascolo Metaverse, which we're very proud of, which involves Lorenzo Leggio's preclinical studies. Leandro is now a Stabman Fellow and an NIH Distinguished Scholar. We have a robust collaboration with Nora Volkoff on a whole variety of things, none of which I talked about today, but another day. And our outside, a lot of within the NIH collaboration as well. We just really are having a lot of fun at NIH. And our very special outside the NIH clinical translation collaborator is Barbara Mason at the Scripps Research Institute and her alcohol center there. Thank you very much.
Thank you, Dr. Koob. I think all of us have been convinced that hypercafia dominates the process of the addiction. So I think we can take some questions from the audience. Does anybody have any questions that we'd like to ask right now? Please go to the microphone and speak your questions.
Thank you for very interesting presentation on such important topic. I have one question. There are pretty novel preclinical data suggesting that there is involvement of fibroblast growth factor 2 and specifically FGF2 receptor 1 and monoaminergic system that they play a role. May you please comment on this?
Yeah, we're following those data very closely because that target could also help with liver disease. So if we had a double whammy where we were actually treating alcohol use disorder and liver disease at the same time, you can imagine the significance of that. So yes, very interesting. But I haven't started any work in our own laboratory on that yet.
Yes, thank you for your questions. So other questions? Yes, please.
Dr. Phillips.
Hello, sir. You've made a wonderful argument in favor of the between systems adaptations. What about the within system adaptation, specifically with respect to dopamine? Do you have any evidence that that might contribute, not explain everything, but also be a factor in all of this?
No, I mean, I think it's a major factor. And I think, you know, there's a study by Pizzagalli, I'm sure you're familiar with, where they've actually demonstrated now this hypohedonic response in humans and shown brain imaging data to support it. So I would argue that it's part of the big picture. And in fact, I think probably dynorphin is partially driving that within system neuroadaptation because, you know, kappa agonists suppress, as you know well, dopamine release in the nucleus accumbens, some of the early work of Tony Schippenberg. So, you know, I think there are many ways to get to Rome. And, you know, I just would like to see us broaden our perspective and engage some of these targets because I think some people enter through some of these targets differently than others. It could be a genetic predisposition. What if you're born with high dynorphin in your shell of your nucleus accumbens? I mean, I'm just speculating.
Right. I was drawn to this question because even in your own presentation, you're showing this hypodopaminergia occurring during withdrawal. And some of our recent work is showing that if you can reverse that, you can also reverse some of the withdrawal signs. So as you point out, there's a role for everything here, but you've done a beautiful job. You didn't give us a lecture today. What you did is distill, I think, your latest textbook. It was magnificent. Thank you very much.
Thank you, Tony.
Yeah, it's about the time to conclude this session, but we have to memorize this new word, hypercafia. And no matter the reviewers will say that it's a tongue game or something. So thank you very much for your joining and participation. I think Dr. Koob, we're gonna take any extra questions if you are interested in. So thank you for your participation again.